Hello, and thank you for your interest in DARPA. My name is William Regley. I'm the Acting Director of the Defense Sciences Office, and I wanted to take this opportunity to provide a brief introduction to our agency and to the Defense Sciences Office. DARPA has been around since 1958. We're an agency of the United States Department of Defense, and the mission of the agency is to, quite simply, create and prevent strategic surprise. The Defense Sciences Office is one of 6 technical offices within DARPA, and we focus on the frontier of science and engineering, both in the United States and globally, looking to harvest ideas that are bubbling out there, perhaps ripe for disruption, ripe for creating something surprising. So we like to think of the Defense Sciences Office as the first line of defense against scientific surprise. Our program managers are constantly out trying to harvest ideas in the science community, engineering community, both industry and academia, looking for things that if we put an investment in the right place, we might get some surprising opportunities for our nation. The present organization of DARPA is in the form of 6 technical offices. And I'll start from the left. The Biological Technologies Office was founded in 2014, and its focus is on, as the name implies, biological systems and biology as technology. Their areas of inquiry right now are infectious disease and how to outpace it, neurotechnologies, and how to use biological processes for manufacturing and other kinds of engineering activities. The Defense Sciences Office, which we'll go into in a little more detail in the next few slides, has several cross-cutting themes that really underpins the entire agency scientifically. We have people investigating mathematics, modeling, and design, physics and materials, social science, and what we call human-machine systems, ranging from work in human-computer interaction to the effectiveness of human teams. The Information Innovation Office is focused on computing software systems and the development of computer systems in areas such as intelligence, big data, cybersecurity, and analytics. A lot of activity within the agency focused on cybersecurity, for example, is housed in the Information Innovation Office. The Microsystems Technology Office was founded 25 years ago out of the Defense Sciences Office. And its mission is on electronics, how to make electronics cheaper, how to make them consume less power, how to make them more reliable, how to use them for information extraction, whether it be signal processing or imaging, or perhaps in cameras or other kind of sensor devices. Then the last 2 offices, Strategic Technology and Tactical Technology, are platform offices. Strategic Technology Office focuses on networks, communications, electronic warfare, position, navigation, and timing. Usually large information and computing and communications networked systems of systems. The Tactical Technology Office you can think of as the platform office. Many of the programs in the Tactical Technology Office take the form of an embodied system of some sort, whether it be the DARPA Robotics Challenge, where we were looking at search and rescue robots, or the active unmanned naval vessel. They focus on platforms across the domains of air, space, ground, and maritime, both surface and subsurface systems. At DARPA, our programs are centered on program managers. These are the individuals that drive the agency and develop the programs that we solicit your efforts for. So when you think of interacting with the Defense Science Office with your ideas, these are the individuals who are right now cooking the next generation of projects that the agency will pursue. Typically in the Defense Science Office, we do between 8 and 12 major programs each year. For information about these program managers and what they may be interested in, you can look at our website, darpa.mil, and pull up their biographies and understand what their interests are. If you are working in areas of science and engineering that you think touch on the interests that are being pursued by our program managers, you should email them or reach out and find out if there's an opportunity for you to contribute to the things that they are developing. Within defense sciences, presently, we view our portfolio in the terms of these 4 areas. On the left, mathematics modeling design cross-cuts almost everything we do. We have foundational programs in mathematics, in simulation and modeling, as well as in various aspects of design, both mechanical design, human machine systems design, and design theory. Physical systems includes work in physics, quantum, position navigation and timing, and materials. This is really the part of the DSO portfolio that has been there the longest, going back to the 1960s. More recently, we've looked at human machine systems, the idea that humans and machines together are more than either one if on their own, and understanding how we can make more effective use of human machine teams. Social systems is an area we stood up about 2 years ago, where we looked into how to pursue questions in social science and methodology for doing social science. To give you a sense of the diversity of the programs in our portfolio, these are some of the recent solicitations over the last year. You'll see everything from photon detection technology to young faculty awards to gamification of idea generation. So it's a very diverse portfolio. And if you're working in areas that might touch on program interests that you see, again, we'd love to hear from you. I hope this introduction to DARPA and the Defense Science Offices was useful. Again, if you're working in areas that you think could contribute to our mission or the interests of our program managers, please don't hesitate to contact us. We look forward to hearing from you.
The most important advice I can give is to read the BAA. The program manager and his team have carefully constructed the BAA. Words in the BAA are meaningful. Your proposal must address the must and shall requirements contained in the BAA. There are should requirements that also should be addressed. I'm going to provide a roadmap through the BAA that you can use in your proposal preparation. First, teaming is strongly encouraged. Cross-disciplinary teams are anticipated per BAA Section 1B. Proposer profile list can be found at BAA Section 8B. Your proposal will contain 2 sections, administrative and technical. See the mandatory templates for these sections referenced in BAA Section 4B2. The technical section of your proposal will constitute Volume 1. Doing well on this section could lead to a selectable determination in accordance with BAA Section 5B. Note the particular technical section requirements in BAA Sections 1B through 1D. The Technical Management Proposal Volume must address BAA Attachment 2 and Attachment 5. Volume 2 of the proposal will be your cost proposal. Doing well on this section could lead to contract award. Your proposal must comply with BAA Attachment 3 and Attachment 5 cost proposal requirements. Volume 3 of the proposal must address the administrative and national policy requirements. This would be in accordance with BAA Attachment 4. Proposal Preparation Submission Requirements. Instructions for proposal preparation are detailed in the BAA and its attachments, as posted on fbo.gov. The RadioBio BAA number is HR0011 17S0021. You should follow the instructions closely. All questions regarding the BAA are to be emailed to radiobiobaa@darpa.mil. Frequently asked questions, including today's, will be available on the DARPA DSO Opportunities website, identified in Section 8A of the BAA. You should read this regularly as it is updated. Funding instruments resulting from the BAA will be procurement contracts, cooperative agreements, or other transactions. You must identify which of these instruments you desire in your proposal. Abstracts in advance of proposals are strongly encouraged. This is addressed in BAA Section 4. See the mandatory abstract template at BAA Attachment 1 as referenced in BAA Section 4B1. You must assert rights to all technical data and computer software generated, developed, and/or delivered to which the government will receive less than unlimited rights in your proposal. This is addressed in BAA Section 6B4. The assertions will apply to prime, contractor, and subcontractors. You must use the defined basis of assertion and rights category and justify the basis of assertion in your proposal. Abstracts are due no later than March 7th, 2017 at 4:00 p.m. Proposals are due no later than April 12th, 2017 at 4:00 p.m. Proposals will be evaluated against the criteria found at BAA Section 5. The criteria in descending order of importance are overall scientific and technical merit, potential contribution and relevance to the DARPA mission, and cost realism. These criteria are defined at BAA Section 5. The government reserves the right to select for award all one, some, or none of the proposals received. The government anticipates making multiple awards. Since there will be no common statement of work, proposals will be evaluated against the criteria in the BAA based on their individual merit and relevance as it relates to the stated research goals and objectives. As previously stated, read the BAA. It contains the information you need to prepare your proposal.
Good afternoon. My name is Mike Fiddy. I'd like to introduce you to a new DARPA DSO program called RadioBio. The goal of this program is to determine if purposeful signaling exists between biological systems, and if it does, to determine what kind of information is being transferred. The objective here is to see if, in parallel to the better understood chemical signaling, electromagnetic waves might also be involved. This is a basic research program. It has very challenging goals, and we're definitely interested in very speculative ideas that might be forthcoming related to this overall program goal. A lot is already known, of course, about the role of electromagnetic waves in biological systems. Here you see the entire electromagnetic spectrum laid out, low frequencies on the left, high frequencies on the right. At the low frequency end, there's a lot known about neuronal signaling, magnetic navigation, and some speculative results on signaling using low frequencies in biofilms. At the high frequency end, of course, we're familiar with photosynthesis, vision, bioluminescence signaling. In the middle region, the gray region, which covers many orders of magnitude of the electromagnetic spectrum, then over the last 30, 40, 50 years, there have been many papers published which show some observational evidence of effects occurring in this frequency regime, but no definitive evidence of purposeful communication occurring. The goal of the program is really focused on understanding whether in biological systems, whether they are single cells, clusters of cells, collections of tissue, structures that are formed from biological materials, whether these structures are capable of serving as components in an electromagnetic transmit-receive system. The interest is in whether these structures can behave as antennas, if they are antenna-like, and if the fields from these antenna-like structures, either near field or far field propagating waves from them, could be transferring information. The assumption is these would be tunable resonant structures, and that any communication based on electromagnetic fields involving these structures would be relatively low power. We're not interested in this program in high power interaction of electromagnetic waves with biological systems in which clearly heating occurs, leading to some biological effect. To date, this grey box in the middle is possibly the part of the electromagnetic spectrum in which there is least known and much more to be understood. And we're looking to have teams form in which definitive evidence can be found based on modeling predictive quantitative models of whether or not there is any signaling or communication occurring between biosystems. We're not excluding by any means studies at the low frequency end or the high frequency end, but we would definitely be interested in seeing proposals in which frequencies between kilohertz and tens of terahertz possibly being involved in signaling are studied. In order to have definitive outcomes as to whether signaling occurs or not from this program, we're trying to construct it in a way in which we avoid theoretical work being done which can't be tested, in which models are developed that are not parametric and don't provide, if you like, a continuum of responses that allow us to better understand mechanisms that are involved in signaling. And we want to avoid experiments, of course, that have poor controls, are very difficult to verify. Reproducibility of experiments, experimental results under a variety of different conditions is very important, which is why building teams that can bring together many of the skills required to address these issues is very important. So we're looking for predictive theoretical models, not observational research. We're looking for those models to be able to predict in a quantitative way, for example, field strengths, bandwidths, channel capacities, that then allow us to do much more precise and parametric models to test those theoretical models and give us a deeper understanding as to what's actually happening. And all experiments need to be controlled, recognizing that the environment in which any biological signaling may occur is very noisy, a lot of background noise. There could be, of course, many differences between species, um, environments, all those factors that could and are known to make experiments in biology so difficult. In this program, we need to be clear to define all of those possible extraneous parameters and understand their importance and know that they're being controlled in order to have confidence in the results obtained. Why are we doing this now? Well, there are a number of reasons. One is the increasing number of papers reporting observational effects across the entire electromagnetic spectrum. The improvement in equipment for making very sensitive measurements, narrowband tunable sources and probes that could more precisely determine field properties in cellular environments, and the greater availability now of molecule cells, cells in colonies, the biological structures that could be the basis of a proposing team's study to see whether communication goes on or not. We believe that although the experiments are very challenging, the modeling of the structures as possible antennas responsible for transmitting and receiving electromagnetic waves is probably even more challenging, since these structures are time varying. They are almost certainly a lot smaller than the wavelengths which may be involved in signaling, and the environment, the medium in which the electromagnetic waves are transmitted, the channels involved are highly complex, lossy, scattering, all of which are factors in terms of defining how effective an electromagnetic signaling mechanism might be and over what range, if indeed signaling does occur. Why is the bioantenna modeling so difficult? Well, if we look at traditional antenna design, one's normally interested in a rigid structure which is broadcasting into air. We're normally looking at propagating waves in the far field from that structure. And an ideal antenna would have a length proportional to about half the wavelength of the operating frequency for that application. As one makes antennas physically smaller than a half wavelength, then a number of things happen. One is that the Q of the antenna, which is a measure of the energy stored per cycle of the field, or equivalently the center frequency of the antenna divided by its operating bandwidth, that Q factor increases dramatically as antenna get a lot smaller than the wavelength. So the efficiency of it as an antenna decreases. The field surrounding the antenna in the near field can be very large, which is why we're interested in exploring near field effects. And the fact that if structures, biological structures, are behaving as antennas. They are likely to be time varying. This introduces an interesting new dimension to the modeling of antenna performance. They are broadcasting into, excuse the word soup, but very complex media. Oftentimes the permittivity, permeability, conductivity, and the frequency and temperature dependence of these materials, the environment in which the antenna finds itself, and the materials from which the antenna itself might be made, all have to be characterized and are variables themselves. And then lastly, in the case of ideal antennas or bioantennas, we shouldn't exclude the possibility of arrays, uniform, non-uniform arrays of transmit-receive components in the biological case, perhaps serving a role to improve directivity or gain if purposeful signaling is truly happening. So this is a difficult problem, and I'll come back to it again in a moment. We believe that to address this goal of purposeful signaling and reproducible results and predictive models, we need a team that exhibits strength in the 4 areas shown here. The top 2 in blue, modeling and theory, will require in the team that a hypothesis can be made of signaling channels that might exist. Could be more than one hypothesis put forward, in which it's highly possible that an electromagnetic wave rather than a chemical communication process occurs. And from that hypothesis and identification of the structures involved in the biosystems under consideration, a model for those structures as antennas, understanding the energy source driving them, the Q and the possible range over which they could broadcast can be calculated. And predictions can be made, which for the lower block, experimentalists can work towards confirming or not. So the biological structures associated with this hypothesized signaling channel will be provided in a reproducible way, thoroughly documented, and an environment for them that's controlled, in which possible sources of noise for electromagnetic waves over the frequencies of interest can be controlled, allowing experiments to be done to confirm, as a result, for example, of some stimulus to the biosystem, which might be expected to trigger a signal, whether those signals can indeed be found and identified, and done so in a reproducible way. So in order to ensure these goals are met, the program is divided into 2 phases. Phase 1 is split into 2 parts. Phase 1A is a time during which the hypotheses are defined and refined, and the biostructures involved and the tools required to probe whether electromagnetic signaling goes on at the expected frequencies for those structures will be defined and demonstrated, so that at the end of phase 1A, at the end of 12 months, we can be confident that everything is in place to conduct a series of experiments to validate that hypothesis, confident in the knowledge that it should be possible, if indeed signaling occurs, to make measurements that confirm or come close to confirming the predictions of the theory. So by the end of phase 1B, we would hope to have identified a set of hypotheses for which there appears to be strong reproducible evidence that some electromagnetic signaling occurs. So phase 1 is the discovery period. Phase 2, which is a lot more flexible, will build on the identified signaling channels from phase 1, and they will then be probed more carefully. The models will be refined, and as I'll mention later, convergence between predicted and measured parameters will allow us to better refine the model and understand how applications associated with that specific signaling channel could be applied perhaps to teach us more about communication in complex environments, or provide applications directly targeted towards a biological function. So the phase 1 milestones are listed here. This is all in the BAA. As I mentioned, multiple classes of hypotheses are fine, and that some may fail to be validated is fine as well. We will learn something from that. But very important is that at the end of the first 12 months, we have confidence that the model for the hypothesis and all the tools needed to test it are in place. If this is not convincing at the end of 12 months, then some redirection of the effort towards a more promising approach may be necessary. Because at the end of phase 1, at the end of 24 months, we want to be sure that tests are completed, reproducible, and tell us one way or another with confidence whether a signaling channel is being exploited or not for some biological purpose. Phase 2, to repeat, is very open. Each class of hypothesis that has been validated may have its own unique set of applications. Again, then the first few months will be to identify the tools necessary to exploit that electromagnetic channel, perhaps for direct regulation of a biological process, all the tools necessary in order to make the measurements that verify and close the gap between the theoretical predictions and the measurements need to be in place after 36 months. And the final year of the program will be to investigate exploitation of this communication channel, verify that it's a mechanism which perhaps occurs more broadly and is robust to different environmental situations, that others, possibly other teams in the program, can also follow the procedures developed and reproduce those same results. So the technical areas for this program are seen as being two. The first really being the technical area in which we expect to see the most proposals. So this is the hypothesis testing, technical area 1. And the 4 components I mentioned previously of theoretical expertise in bioelectromagnetics and antennas, and experimental expertise in preparing biological systems for the experiments and then actually conducting the experiments. These teams will be addressing the complete cycle of hypothesis, prediction from modeling, and verification or not from experiments. We understand that there could be groups, individuals who have a lot of expertise in theoretical modeling of biosystems and or expertise in antenna design. And so we, in technical area 2, invite proposals that are purely theoretical or numerical modeling in nature, and that might have approaches to understanding how transmit, receive antenna-like structures in biological systems could occur, could possibly be very low frequency, high frequency, or surprisingly broad band. And this theoretical support may or may not fit with one of the technical area 1 teams and their particular hypotheses. If it does, then of course we will bring those teams together to have the theory team support wherever possible a technical area 1 team. But it's also possible that the tech area 2 teams will simply support the overall goal of the program without, at this point, necessarily being able to connect with an experimental team capable of verifying or deriving any experiments from their predicted models. Very important throughout the program for both TA1 and TA2 teams is that metrics are identified. The metrics are up to the proposers to identify, but it's very important that metrics are identified in order to show that the hypotheses, the theory and the experiments can be definitively quantitatively tested. So a list of metrics which also can identify a precision for each with which one might be able to theoretically predict or experimentally measure those parameters is important, so that as time goes on, every 6 months, every year, this metric timeline can be reviewed, and the gap between the theoretical prediction and the experimental measurement can be reduced as convergence occurs between our understanding of what might be going on in terms of the model, and improvements continue to be made in terms of the experiments that we do. So for example, one of the antenna parameters was the resonant frequency. It might be that early on in the program, one only observes a correspondence between the predicted resonant frequency of a biostructure antenna and the measured frequency to be they may be a factor of 2 different. But after, say, 18 months and 36 months, we might hope that the difference is reduced to less than 20%, maybe less than 10% towards the end of the program. Similarly, with identifying the material properties of the media in which the antennas are radiating or receiving, they may differ in terms of model and experiment by a factor of 5 initially, but as we learn more, then the convergence between the two may drop to 10%, maybe 5% by the end of the program. So in the executive summary of a full proposal, we're looking for a couple of tables to be completed. 2 in the case of Tech Area 1 submissions, and the one seen here for Tech Area 2. We would like a concise summary of the modeling strategy for the biological structures that could be involved in the electromagnetic signaling hypothesis that's being tested, and the strategy for the design and prediction of the characteristics of the associated antenna. A brief description with a reference made to where in the full proposal more details can be found. Tech Area 1, the teams involved in doing experiments. This table looks a bit more intimidating, but is still pulling material from the full proposal and summarizing it in the executive summary to identify what are the 1, 2, or more hypotheses being tested, what are the biosystems involved, what are the frequency ranges likely to be involved in a purposeful communication channel, and so on. So this will focus and summarize in the executive summary the particular approach any one team will take. Now, teaming, we realize, could be difficult for some, so we do invite those who might like information about other possible proposers. We invite them to submit a one-page profile which includes contact information, brief description of technical competencies and interests, and if there's particular expertise needed from other organizations or teams, to include that. There is a deadline for the submission of this to RadioBio BAA at darpa.mil of 2 p.m. on February the 24th. This is entirely optional, but if you would like to engage in this teaming exercise and submit this one-page profile, then we will consolidate them and send them out to everyone, and then you can contact individuals, groups as you see fit to help build the team that you might be looking for. All the details about the abstract can be found in the BAA and should be submitted to the same email address. The abstract needs to list what the hypotheses and the biosystems are likely to be if you submit a full proposal, what you plan to do, why you think it'll be successful, and what kind of milestones, risks, obstacles you might face. And also capabilities and management plan, a brief summary of the team, understanding that teaming arrangements may not at all be final, but to give us some idea of how you're thinking about structuring a team and organizing the team, and a brief estimate of what you anticipate the costs to be for phase 1A, 1B, and phase 2. We hope that you will submit an abstract, and we look forward to providing feedback to you based on that abstract, in particular related to the size and level of effort that you are suggesting. So to summarize, the name of the program is RadioBio. All correspondence, all questions you might have need to be submitted to radiobiobaa@darpa.mil. So just to summarize one more time, the goal is to determine if purposeful electromagnetic signaling occurs and what information is being transferred. We're looking for hypotheses where electromagnetic signaling may well be occurring, and we're looking for evidence, experimental evidence and predictive models to validate, verify these hypotheses. So more than one hypothesis is fine. We need to understand the signaling process. We need new types of models for bioantennas to explain if signaling occurs, how waves are transmitted and received. And then we need experiments to be conducted for TA1, which are totally transparent, consistent, reproducible, fully documented, to be sure that other groups are able to observe the same phenomena. And then towards the end in phase 2, we're hoping that if some electromagnetic communication channels are verified, that we can start investigating them, learning more about them, both for biological applications, but also for communication applications in general. So with that, I will end, and I look forward to receiving abstracts, questions, and eventually proposals. Thank you for your attention.